Hi, and welcome to today's webinar on how to analyze the human transcriptome array. Today we'll be concentrating on splice variant analysis. The human transcriptome array analysis workflow consists of basically two different software packages. The first is Expression Console, which is used to produce the signal summarizations and also perform QC of the arrays. After you've identified the arrays which have passed the QC metrics and you're comfortable with doing further analysis on, you load those into Transcriptome Analysis Console or the TAC software. There you can do either gene level analysis, exon level analysis, or alternative splicing analysis. Today we'll be concentrating on how to do the alternative splicing analysis and visualizing the splice analysis results in TAC. The alternative splicing analysis is done for each condition pair. So within the Transcriptome Analysis Console, you can set up conditions that you want to analyze. And this is described in another webinar, which is available on Affymetrix.com. So once those are established, the gene level analysis proceeds, and there'll be some metrics that are calculated at the gene level. This includes the average signal and standard deviation across the gene for each condition pair. And then for the pair, there will also be a gene level fold change calculated. Then at the exon level, there's a calculation performed to calculate the biweight average, the standard deviation, and the normalized average signal for each probe set in each condition. And then between the probe sets, both the fold change and the splice index for each probe set is calculated. A one-way ANOVA is calculated to produce p-values for each of the conditions, and then the FDR correction that is applied to all the p-values. If you have questions about how to set up an analysis and further details of this, please refer to one of the other webinars on the Affymetrix.com. The alternative splicing results are broken up into three main pieces. There's a summary page, which consists of the breakdown of the number of expressed genes in each condition and the number of genes with an exon or probe selection region, also called the PSR, with a splice index of greater than 2 or less than -2, which basically corresponds to a two-fold change. Second, there's a results table. It has two halves. On the left half is sort of gene level information. On the right is the exon or junction level results. And these results at the exon and the junction level are grouped by gene, so they travel sort of as a pair or as a set when you do the sorting and stuff. And I'll demonstrate this in the software when we get there. In addition, there's a gene level annotation information provided, including gene symbol, position, and a short description. Then there's also the transcript visualization piece, in which the exons and junction probes are visualized and are color-coded based on their expression or their splice index. The default filters in the TAC software are to identify things with a splice index greater than 2 or less than -2, again corresponding to about a two-fold change, and then FDR corrected p-values of less than 0.05. So now we'll transition to the actual software so you can see what the analysis results look like and go through some simple analysis there. When you first launch TAC, this is the page that you're landed on. In previous webinars, I've described how to actually set up an analysis. Today we're going to go to the Open Existing Analysis, and we're going to open one of these splicing results file here. Cruise over here, maximize the window. So this is the view after the results are loaded. On the left half, by default, at least in the split view, you end up with the summary, and on the right half is the visualization. I'm going to close the split view just for a second so you can see. Here you can go up to the split view to turn that off. Here's the summary information. It contains at the top basically a description of the experiment that's being performed and a summary of what was found. So we looked at a total of 67,000 genes here. The results are then displayed, for the comparison between the muscle and the liver genes. And then there's a breakdown of the number of genes that are expressed in both conditions. They're expressed in muscle and not in liver, and vice versa. And then here is the first set of information that's sort of relevant to the alternative splicing. So there's basically about 9,000 genes in this case that have at least one PSR or probe selection region with a splice index greater than 2 or less than -2. This is for the coding genes, and then you can see down here we have done a similar breakdown of stuff for the non-coding genes. And so in that case there's about 1,300 genes with a splicing event in it. A little bit further down we have the algorithm options. If you're interested in explanation of the algorithm options, I would refer you to the other Introduction to Splice Variant Analysis webinar, in which the rules and the interpretation for what these various things mean has been explained a little bit further. here is once again a restatement of the default criteria and the conditions that were identified. If we now click on the table view, this is basically a simple table of all of the data for this experiment. We have the gene annotation information, we have the gene level information here on the left side of the table, and the exon or probe set information here on the right side of the table. Columns can be reordered by dragging them over, and then they're inserted. And then if you want to, you can change the sort order or the filters basically by right-clicking on a column header. So I'm going to change the sort order of this column to sort by ascending. And this will take a quick second here. Now the one thing to remember is that the data is actually organized by gene. So if we look here at the results when they finish sorting, we'll see that here in the splice index column, this is a minus 11.7, 10.8, and then here we have 11.69. That's because what happens is it goes through and finds all of the individual PSRs or junction probes and finds the one with the lowest splice index in this case and puts it first, and then identifies that gene and then finds all the other PSRs and junction probes for the gene. And so it's organized basically by gene. So these are all the junction and PSRs associated with RGMA here in ascending order, and then followed by next by MICAL2, and again they're ascending. Okay, so it's not simply a sort ascending on the column, it's a little bit fancier sort by the column with respect to gene. Okay, so in addition, if you have more than one comparison, you can choose your comparison up here. You can choose to show and hide some additional data columns. And so first are things that are relevant at the gene level, and then further down here we can find here's some annotation information if you wish to display that. And then here's two columns that I'm going to add, which we'll come back to later, and that is for the muscle and the liver PSRs or junctions, whether they were considered to be expressed or not. So I'm going to add those two columns. Click back and we can see these two have been added here. I'm going to reorder this so that the splice index is here. And the reason I'm going to do this is for when we do the split view, in order to sort of see the table and the transcript and genomic view at the same time, I'd like to be able to minimize this so that there's more room for display. And so I'm sort of setting myself up for that now. In addition, I'm going to filter this column just to make it a little bit more clean. I prefer to just have the PSR data displayed in the table, which makes it a little bit more compact and a little bit easier to view. So now let's go ahead and you can search for a particular gene of interest. In this case, I'm going to look for annexin A7, and then we hit the next button, and it will then search this. It's possible to do a substring search is what it's called, or if you just type in a portion of a name, it will search through and find just all the possible matches for it, and then you can navigate to the next or the previous one using these buttons. Okay, so now we've navigated to annexin A7. One thing to note is if you search for a gene symbol and nothing comes back, it's possible that it's just not in the table, that it's actually in the data but it doesn't meet your filter criteria. And so you can adjust the criteria over here by turning on and off various checkboxes, or you can actually click the Show All button. It's not recommended to do that because you'll load lots of data, most of which is not relevant for the splice index comparisons. But if you're interested in what happened to your favorite gene and you can't find it in the table, that's how you go about doing that. Okay, so now let's go back to the split view. And you'll note that even though I searched for the annexin A7, which is highlighted down here at the bottom, the gene that's displayed over here is MICAL2. So let's go ahead and you can change this either by double clicking in a row to update the transcript visualization, or up here at the top you can do the View Selected Gene button. And so now what's displayed on the right is actually the gene annexin A7. Okay, and so now I'm going to set this up a little bit, modify the table view over here a little bit so that it's a little bit nicer and I have a little bit more room to dedicate to the transcript view. It's possible also to do the split view where the table goes on the top and the visualization goes on the bottom, and so that is a preference depending on which way you prefer to see the data, and that's generally driven by, do you have lots of transcripts? If you have lots of transcripts, you probably want the split view this way. If the transcripts are very long with lots of exons but not very many, then you may want to do it the other way. So I'm going to do it this way The other thing I want to do is further setting up my table. I prefer to have gene symbol, then the gene fold change visible for me, just so that I have some idea. We'll scroll those guys over here to the edge. And then resize the splicing index a little bit. Oops. And these other two columns that I mentioned before, which we'll come back to, and then we can drag this over. See, so now I can see all the relevant information for me over here in the split view, and I have my transcript view maximized over here. So the first thing I'm going to do, because I'm colorblind, you can see that we're in the standard splicing index color coding, goes from green to red, and I don't like that, so I'm going to click the down arrow and I'm going to adjust this to be this light blue color. So now down regulated is light blue through to red, and this is the splice index of 4 and -4, and here's an intensity color scale. Okay, so what we have here is a little sort of control or navigation box here. On the top is the structure view. I'm going to click the zoom to fit button to maximize this. Okay, and it's basically each exon is the same size, and then the junctions are displayed as these little boxes that go over the top, right? So this junction, by mousing over, you can see it connects this exon to this one, and here's an exon below it. Okay, and so this is an example of a classic cassette exon, which, if you're not familiar with, I again refer you to the Introduction to Splice Variant Analysis. But basically, it's a condition where this exon is either present or not present in the transcripts and sort of spliced in and out. And so one thing here, this is just basically the exons are all the same size, the junction probes are indicated above them, and it's color coded by the splice index. And so if we choose to, we can go ahead and we can draw a box in order to zoom in just this section of the transcript. And this is in the structure view. Similar functionality exists in the genomic view, which is on the bottom, but we're just looking at the top. And so what we can see here right now is we have this exon, and the two junction probes to the left and the right of it are all overexpressed or expressed higher in muscle. And then we have this junction probe, which is a little bit overexpressed in liver. So now, if we want to actually check and look at the underlying expression values that we're seeing in both liver and muscle, we can go up here in the structure view and we can open these two pull downs, if you will. I'm going to go back to the zoom to fit to sort of resize it, and you can now see the full view of the gene again, and you can see the blue is relatively high expression and the yellow is lower, and we can see for this exon that we have, you know, a average signal here of 7.3, and this is the average signal of 3.72, so it's like twice as much. Okay, so this is a higher expressed in the muscle than in liver, right? And so then if you also mouse over here, you can see what the normalized expression for each of them are, and then the splice index, which is calculated using the normalized values. So here we have a classic example of a cassette exon. Okay, so this is one view at the top. I'm going to go ahead and close back the raw signal values and leave just our fold change. Okay, and then down here at the bottom, we basically have the genomic view. So it's possible to do two different ways to look in the genomic view. The first is if you toggle this box on and off, you get truncate the intron exon regions. And so what happens here, this is sort of a little bit more familiar view of a gene that you would see sort of at UC Santa Cruz or in other genomic browsers. But you can see when you've got it in this sort of view, it's a little bit harder to see the color coding associated with each of the individual exons. So we've introduced a way to actually truncate the actual intronic regions, but leave the exons sort of relative sizing available so that this exon here is actually larger than that one. So the exons haven't been scaled down to minimize the data, right? So what we've done is basically minimize the intron so that you get a more compact view of the gene. It's possible by clicking on in this row here or in here to be able to select the row, to be able to highlight and see where it is. Okay, note also that when you select an exon here or a PSR, it's also highlighted in the table for us over here. So we can see right here the splicing index for this guy is 11.28. If we mouse over it, we'll also see that it's 11.28. So you can get that information one of two ways, either by clicking in the view or looking at it in the table. And if you want to see which exon corresponds to this PSR, you can click on it and now it's highlighted here for us. So we'll go back to the classically expressed cassette exon. In addition, you can see here on the left we have transcript identifiers. Right? If you right click on them, it's possible to copy that identifier out, or you can link out to either Ensemble or NCBI depending on which piece of information is available for which transcripts. It's also possible, if you want to simply look at transcripts from Ensemble, you can choose to just view the Ensemble transcripts. The other ones remain here, but you can see it's basically a double sort that's happened. First we've sorted within the Ensemble transcripts and sorted secondly within the non-Ensemble ones. You can also do the same thing for RefSeq, and the third is Other, which includes identifiers from places such as UC Santa Cruz. And go back to the default view. So this is a relatively simple case just to sort of demonstrate the overall functionality within the genomic view here. So now if we go back to our table, and we'll just zoom up to the top of the table, and we'll look at this MYB PC1, which is a somewhat semi-random selection of a gene that's a little bit more complicated than the annexin A7 is. And so in order to get that to show up, we double click on the row itself. So now you can see here why I've actually set this up the way I have, because there's a lot more transcripts, and so I want to maximize the real estate so that I can see them. And so you can see that we have a little bit more going on in this one. Okay, so we have a bunch of things that are blue, which means that they're higher expressed in liver than in muscle. We have this guy who's more expressed in muscle than in liver. Okay, and so if we drill into here, let's look at this guy first because it's 1. You can zoom in and we can see that we have sort of the equivalent of the classic cassette exon, right? This is higher expressed in the muscle. This is higher in liver with the junction overhead. And so that looks pretty nice. Okay, so now one functionality that exists within TAC is the ability to sort of sort the transcripts to try and get them oriented relative to their expression value. So if we take this exon, for instance, you can see some of the transcripts contain this exon and other ones don't. So what we can do is if we highlight this guy, we can choose to move all the upregulated transcripts with this exon at a higher level of significance and overexpression will go to the top. So we'll choose upregulated to the top, and you can see now that all the transcripts that contain this exon, as having it red, are at the top of the list, and those that don't have it or that would have this exon in blue go to the bottom. So this is a simple way to start analyzing what transcripts might be present in which sort of tissue. So if we know that this is red, which means it's overexpressed in muscle than in liver. So if we want to look at what are potentially the transcripts that are expressed in liver, so we could say that, oh, so then if this is overexpressed in muscle, let's look at the ones that don't have this as red or would have it as blue. And so that brings us to these 4 transcripts down here at the bottom. Now, one thing that would be interesting is, do we know any information about whether this exon itself is actually seen in liver or not? And so that's why I added these 2 additional columns over here. So it's the muscle PSR expression and the liver PSR expression. And so basically by looking at the DABG value, or the data above background value for each of the PSRs, there's sort of an estimate of whether this PSR has been expressed over background, right? And so for this actual exon, so let's select it. So we can highlight it in a table, and it's actually scrolled off the bottom here, so we'll scroll up so we can see it. And so it's this blue guy here, and we can see that 4 out of the 4 samples had this exon detected in muscle, and 0 out of the 4 had it detected in liver. So that would suggest that our liver would only contain transcripts from these 4. But so let's look at this a little bit more carefully. If you look above, there's also some blue exons here, like this guy, that are higher expressed in liver than they are in muscle, but they're not expressed here. So these exons need to actually be expressed in liver in order for them to be overexpressed in liver. So there's 2 possible explanations for this. The first is that while we have 4 transcripts here, there are other transcripts that haven't been published yet in any databases, and so we don't have a representation of them here. So there's a novel transcript out there in the world perhaps that exists that doesn't contain this exon but contains one of these 2 exons or both of them in some combination with the others. The second possibility is that there's actually a very low level expression of this exon in liver. It's just not detected by this. So there's sort of 2 possible interpretations, and further follow-up experiments would need to be done in order to investigate that. So now if we turn our attention to what are the pool of transcripts that would be expressed in the muscle tissue. One way to think about it is you could potentially naively go in and assume that anything that is blue wouldn't be expressed in muscle. That's not quite true, and I can show you exactly why this won't happen, again, using sort of the same sort of thought experiment. right? So if you try and avoid any blue in the transcripts, you can see all of these guys aren't going to be selected. So we could look possibly at this guy and that guy as we move from left to right. They don't have any blue, any significant blue, no significant blue, all the way across until we get to here, right? So when you get to this final set of exons here, you end up finding again some that are significantly overexpressed in liver, but they also contain this exon that's present in muscle. Based on this, we have again two possible explanations. One, that there are transcripts that exist in nature that haven't been put into the databases yet, which is quite possible. And the second is it's just a relative expression, right? So you have to remember that this is a fold change expression. What it means is that the amount of this exon is found relative to the gene level expression in liver is higher than it is in muscle, right? Because we did the normalized expression. If you sat through the webinar on Introduction to Splice Analysis, one thing that was pointed out there is an example in which if you simply have the loss of a deletion, it actually looks like the gain of an exon. And so that would be one possible explanation for this. So we have a couple, again, explanations for the interpretation of the data. And so these kinds of thought experiments need to go on in order to extract the true value from the human transcriptome array and trying to tease out the alternative splicing events that are happening and which transcripts might be more overrepresented in which tissues. Okay, so that is basically a summary of the functionality within Transcriptome Analysis Console. Before I leave you, though, I want to point out a couple of other resources that are available on Affymetrix.com. Okay, so for all of our arrays, there's the NetEffects Analysis Center, which allows you to get more information on the probe sets, the genes that they map to, and the other sort of annotation-related information for them. In addition, there are some manuals for both Expression Console and the Transcriptome Analysis Console, or TAC. There are white papers and other data sheets available on Affymetrix.com. In addition, there are other training videos that have been made, either related to how to do the analysis in Expression Console, how to do gene level analysis console, and how to set up these analyses. And then also, as I mentioned, the introduction to splice variant analysis. As always, feel free to contact the technical support line at the 188 DNA chip or via the email at support@affymetrix.com. I'd like to thank you for your attention.